Hey guys, Merry Christmas coming up. And for those of you in the future, when it is July, Merry Christmas in July, because you need as much Christmas as possible in the sweltering weather of Arizona summer. So, today we're just going to do a really quick clip on the photoreceptors of the eye, rod cells in particular, and the dark current. So basically, what's happening when we're in the dark, what's happening when we're in the light for the rod cells. And yeah, this is, by the way, welcome to my study Nook. My very awesome sister got me that handheld pocket watch. Just, yeah, that's a good person. So, yeah, welcome. Now, let's dive into the eye. So, just look at what is in the blue box at the beginning, okay? Ignore all the rest of the stuff. The easiest way to understand the eye, I find, is to begin with what happens in the dark. So in the dark, something occurs that is called the dark current. Dark because it happens in the dark. Current because sodium ions flow into the rod cell in a current. So it's a dark current, a current that happens in the dark. Now, how does this happen? Well, there's a sodium transporter, a channel, sodium channel that opens when cyclic GMP, this little green thing, is bound to it. So cyclic GMP is the doorman. And in the dark, we have lots of cyclic GMP. And so this is going to be open, and sodium comes in. Okay? Why do we have lots of cyclic GMP in the dark? We'll get there. But right now, know that the dark current means sodium is coming in, and that's the current it's referring to. And when this is happening, that means that the eye is going to send the signal, and we're not going to get into detail on it, but it says, I'm seeing nothing, so don't send any action potentials to the brain. Okay? Okay. Simple enough. Dark current, sodium comes in, cyclic GMP is holding the doorway open. Now, what happens in the light? Now, forget the orange box. Looking down here at the main part of the page, light will come in, shapow, and it will hit rhodopsin. Okay, rhodopsin is our photoreceptor. When light, basically a hydrogen, will hit it, a photon by itself, when it gets hit by it, it actually changes shape. And this comes from vitamin A. It's also known as retinal, A-L at the end. And so when it gets hit by light, it changes shape. That change in shape is going to do something we've seen before and affect a protein called a G protein. You'll notice that I have the letter T in there, however. This is a very specific G protein. It was the first one discovered, and so it got a special name, transducin. It is transducing the signal from a light signal to a protein signal, and so it was named transducin. So rhodopsin gets the light, turns on transducin that's right down below it. Transducin is going to go and do what G proteins do. It's going to turn on some other messenger inside the cell. Phosphodiesterase is going to then grab all of that cyclic GMP that we've been talking about as the doorman in the dark. Well, the doorman gets chewed up by phosphodiesterase. It chews up the ester bonds inside the cyclic GMP. So cyclic GMP is gone, and that means the doorway gets closed. It is really hard to draw in a mirror image. Beautiful. Ah, I should be an artist. All right. All right. So in the dark, it's open with a doorman cyclic GMP. In the light, rhodopsin to transducin to phosphodiesterase, which then chews up all the cyclic GMP, which closes the sodium channel. I hope that's helped. At that point, you've gotten what you need in Q1. There is one other detail that you can completely throw away if you don't want it. Last detail up here, I show that something that is always happening, light or dark, and that is sodium is always leaving. and then potassium comes in and does a horseshoe and goes out through something else. But it's a sodium-potassium antiport, so sodium is always leaving. And so another way of thinking about this is the dark current is when you're balancing the two. And so you have sodium come in to balance the sodium that's always leaving. And then because of that balance, you don't have as big of a gradient, and therefore you're not going to have the signal that says, I'm an active cell, please send stuff to the brain via complicated stuff. Versus if you only have sodium leaving, but you don't have sodium coming in, then you're going to build up a bigger gradient. And then that's going to tell the cell, whoa, I'm actually active. Let's do some stuff, okay? That part you're not going to have any questions on in Q1 Phys. So what for the basics, if you wanted that in-depth knowledge, great. For the basics, just remember the doorman is there in the dark, and he's not there in the light. Remember the proteins and vitamin and proteins in the sequence, and just the name of the transporter. Go ahead and do a recall on the dark current, and then do a recall on the transduction of light, and you should be good. I hope that helps. Good luck on your final, guys, and I'll catch you next time.